broadcast live to you from the Human Genome Project headquarters. The goal of this project is to determine the complete nucleotide sequence of each chromosome through the developing faster sequencing technology.
Here is an example of the whole genome shotgun approach to sequencing. First, cut the DNA from many copies of an entire chromosome into overlapping fragments short enough for sequencing. Second, clone fragments into plasmid. Third, sequence each fragment. And fourth, order sequences into one overall sequence with technology.
Recent discoveries were made at the Human Genome Project headquarters regarding accessibility to bioinformatics information. Gene annotation is the analysis of genomic sequences to identify protein coding and determine the function of their products. Similar to studying a set of genes in genomics, proteomics studies an entire set of proteins that carry out cellular activities. Combined genomics and proteomics enable scientists to utilize the systems biology approach to analyze the behavior of a whole biological system based on the interactions of the parts of the system.
For the topic of the day, we will talk about the size of the genomes. Genome size among the three domains, bacteria, archaea, and eukarya, vary, yet there are general distinct differences between prokaryotes and eukaryotes. Eukaryotes have larger genomes, but fewer base pairs of genes, thus lower gene density due to the activity and presence of the introns. In bacteria, the sequence of nucleotides along a bacterial protein coding gene is not interrupted by a non-coding sequence.
Our next topic is talking about multicellular eukaryotes that have a lot of non-coding DNA. Look how little is actually expressed and coded in this image. Only 1.5% of genes are coded for proteins or give rise to rRNA or tRNA. Pseudogenes make up the non-coding portion. They formerly functioned as genes, but are no longer activated in the transcription process as a result of a mutation and are now junk DNA.
Do you know what transposable elements are? They are parts of DNA that can move from one part to another within the genome, otherwise known as jumping genes. They have an important role in altering the functions of genes or patterns of expression and regulation of generating new sequence combinations.
Recent research has contradicted the perception that transposable elements are known as junk DNA. A recent study conducted by the U.S. National Library of Medicine National Institutes of Health displayed DNA sequences as playing a role in the origin and evolution of many genomic disorders. This disproves the original thinking that it is only known as junk DNA and does not have any effect in the sequences.
Have I ever told you about Barbara McClintock? No? Well, she's an American geneticist who performed breeding experiments with Indian corn in the 1940s and 1950s. She identified changes in the color of the corn kernels and theorized that this was a result of the observed transposable elements on the corn kernels. Transposomes are a type of transposable element that can move by a cut and paste method, which removes the element from the original site, or a copy and paste, which leaves a copy behind. Retrotransposomes are another type of insert, which insert themselves into different locations in the RNA intermediate.
Simple sequence DNA contains many copies of short tandem repeated segments. This is what it may look like. Now we have a question sent in from one of our viewers. Allison from New Jersey asks, what is polyploidy? Great question, Allison. Polyploidy occurs when errors in cell division lead to extra copies of chromosome sets, which may cause sequence changes. Now, looking at the bigger picture,
It's time for evolution and development, comparing genome sequences to provide clues about evolution and development as seen here. The second diagram shows further expansion of eukaryotes through the evolution of closely related species. According to the U.S. National Library of Medicine, homeobox genes are large groups of similar genes that direct the formation of many body structures during early embryonic development. They are present in every human chromosome and appear in clusters as seen in this image. Some homeobox genes act as a tumor suppressant, which means they help prevent cells from growing and dividing too rapidly or in an uncontrolled way. Since homeobox genes are responsible for numerous crucial functions, mutations in these genes result in a variety of developmental disorders.
Thank you for joining us at the Human Genome Project today. We hope you enjoyed learning about genomes and DNA sequencing. Stay tuned for new discoveries on genomes with next week's update.